Welcome back to Drug Diaries, the podcast where we uncover the incredible stories behind the science of drug discovery and development. I'm your host Kaushik Trivedi, and today I'm thrilled to introduce you to someone who's truly at the forefront of making science work for us in ways that are as complex as they're inspiring. Our guest today is Dr. Carly Seshel, a powerhouse in the world of organic chemistry. Now, if you're wondering what that means, let me break it down for you. Imagine being able to design and build molecules that can do exactly what you want them to do inside the human body. It's like creating tiny, invisible machines that can fix problems at the cellular level. That's what Carly does. She's like the architect of the microscopic world, creating the building blocks for new and innovative drugs. Carly's work is crucial because when it comes to developing new medicines, it's not just about finding something that works. It's about designing something that works in the right way, with the right target, and with the fewest side effects. Her expertise is the key piece of the puzzle in turning scientific ideas into tangible treatments that can change lives. And by the way, if you've been following along with our recent episodes, you might remember our conversation with Somesh Mahapatra. We talked about a breakthrough in the treatment of Duchenne muscular dystrophy from a machine learning and AI perspective. Well, today we're going to hear from Carly about her role in that very same project, but from the fascinating world of organic chemistry. Carly, it's such a pleasure to have you on the show. Welcome to Drug Diaries.
Thank you so much. It's really a pleasure to be on here today.
Likewise. Carly, can you share your journey from your academic background to your current role and interests?
Yeah, of course. So I went to college at Williams College, which is a small undergrads only institution. And that's really where I fell in love with organic chemistry and biology. From there, I got my PhD in chemistry from MIT, where I worked with Brad Pensaluti, specifically on the chemical biology of peptides, which we'll get to talk more about today.
Wow.
I now am a fellow at UC Berkeley doing my postdoctoral studies, thinking more specifically about organic reactions that we can do on peptides in order to make them more drug-like.
Carly, tell me more about your childhood and how it shaped you into who you are today.
So I grew up in Southern California. Both of my parents actually are scientists, and my mom was
Wow.
my science teacher in 8th grade. So I was exposed to thinking about how the world works from a very young age. And my parents have always been very supportive of, thinking about nature and health and disease from a molecular perspective.
Right. It's incredible how much inspiration we draw from our parents and the environment that we are brought up in, and that encourages us to pursue the goals and ambitions that are important for us. So I totally get it. And what inspired you to pursue a career in organic chemistry and scientific research?
Mhm. Yeah, so when I was taking organic chemistry in college, it always starts out very challenging when you're thinking about molecules sort of in space. How does this reactivity work? But I do remember hearing a seminar where it really clicked that life and health and disease is written by molecules interacting with each other. And if you can understand how the surfaces of these molecules sort of interact, then you can think about ways to cure disease. And so that's what really got me interested in organic chemistry, especially from a perspective of treating disease.
Oh, that's really fascinating. I think I really like the way you explained the whole concept in such a simplistic manner. That was very helpful. And how have your experiences at institutions like MIT shaped your career path?
They've been very impactful. I think a really special thing about Cambridge and the Boston area in Massachusetts is the proximity to industry. There's tons of biotech and pharmaceutical companies in the area. And so during my PhD, we actually collaborated with a company, Therapeutics, and they're the ones who've had the first commercially available drug to treat Duchenne's muscular dystrophy. And my advisor and my lab mates and I would walk over to the company and meet with their scientists in person. And that really helped us, share information, share expertise, collaborate effectively. And it's tough to have those face-to-face interactions anywhere else.
Wow, indeed, that's a great advantage. And I've been in Boston several times, and it's a hub for the pharmaceutical industry. You've got all big pharma companies there, and partly because of the proximity to MIT, and it becomes easier to get the talent from these institutions.
And there's so many other universities in the area,
Yeah,
all really within walking distance.
yes, it's an amazing city. I love Boston. Kali, can you explain your research on chemical biology of peptides?
Yes. So chemical biology is kind of a funny term because people think it just sounds like biochemistry. But I like to think of chemical biology as you can design small organic molecules that interact and directly affect biology. And so when people think about designing drugs, they're really doing chemical biology. And so I'm really interested in a class of molecules called peptides. And these are like small proteins, but often you can make them chemically just like you would make small molecules. Um, and they're really powerful because they're somewhere in between small molecule drugs like Advil, which is very
Mm-hmm.
small, and large protein drugs like Herceptin, which is an antibody pharmaceutical that's used to treat breast cancer. Peptides, size wise, are sort of in the middle, and so they can be extremely specific to a protein target while also
Right,
being more accessible. You can modify them chemically, and they're a cool mixture between the two.
sure. And what are some of the key findings or breakthroughs from your research?
Mhm. Since you touched a bit on your last podcast with Samesh, I can
Hmm.
definitely start there. But
Sure.
as I mentioned, we had a really fruitful collaboration with Sarepta Therapeutics that in the Pentaludi lab has lasted, I think over a decade at this point. Um, but while I was there, I was really interested in designing and discovering new peptide sequences that could do this delivery. And so really their activity was delivering this large DNA-like structure to the
Hmm.
nucleus of cells. Because that
Right,
drug by itself is effective, it's on the market, but it would really be enhanced by better delivery directly to the nucleus because its
right.
target is RNA, which is inside the cell. Um, and so during my PhD, I took two approaches to find these new sequences. The first was a machine learning guided discovery, which you talked about with Samesh.
Hmm.
And so in our lab, we had a library of peptides with sequence and activity information. And we brought that data set to Professor Rafa Gomez Bambarelli and his student
Hmm.
Somesh. And they designed a machine learning model that was able to predict even better sequences. And the output of that was a list of hundreds and hundreds of peptides. And I picked, I think, 10 or 15 or so to synthesize and test. And many of them were really a lot more effective than what we started with. Um, we were also able to deliver proteins and other types of cargo inside the cell. And so we
Hmm.
found that these peptide delivery agents were pretty, ubiquitous and flexible, which was exciting.
Right.
And we were able to do animal studies in the end with Sarepta. And so
Hmm.
it was exciting seeing those molecules, advance a bit down the pipeline. And I think what we got out of that was a better understanding of specific motifs that can really help
Hmm.
with delivery.
Right,
That was my main takeaway. And then
right.
I'll touch on the second approach quickly, but I
Hmm.
also designed peptide libraries
Hmm.
where it's a mixture of thousands of peptides in solution. And if you treat cells with that mixture, a few of them will be able to get into the cell, and those are
Hmm.
sequences that have high activity. And so in this platform, we were able to fish out those sequences that got into the cell and
Right,
identify them. And so that's a completely de novo approach. It's not really based on any design. And so,
right.
um, yeah, that's sort of an orthogonal approach to the machine learning guided discovery.
Right. So I'm again trying to visualize the whole process and how would that collaboration work between you and Somesh's team. So I'm assuming Somesh and team would come up with a list of peptides which are generated on the AI or machine learning tools, and then they would pass on the list to you, and then you would take that list to the lab and create those peptides. Or do you already have the library of peptides and you just pick the ones that you think would work and just try it on different sort of settings to see how the results are?
That's a really great question. So the awesome thing about peptide chemistry is that it's so modular.
Hmm.
And so if we're presented a list of hypothetical sequences, I did have to go into the lab and make every one from scratch and test every one. And
Wow.
so what that looks like is we use something called automated flow chemistry.
Hmm.
And this continuous flow format is actually a machine that was generated in the Pentaludi lab. And so that's sort of a new technology that allows us to make peptides that are much, much longer than you would be able to make otherwise.
Right.
Um, and when I say it's a modular synthesis, what I mean is every amino acid is
Hmm.
coupled like a building block. And there's some protecting group on the end of one monomer. And so if you couple monomers to each other,
Hmm.
this one has to be deprotected before it can react to another one. And so
Right.
it's a couple, wash, deprotect, couple kind of workflow.
Right.
The end of that is peptide on resin or on a solid support. And then you then remove the peptide from the resin. Now it's a material that you can work with. You purify it. I then conjugated it to the cargo molecule, and then I could do all of the biology assays, so the cell work and such.
Right. So my follow-up question is, how does this automated flow system works differently to the traditional methods?
Yeah, there's a variety of ways that people make peptides. Um, the the oldest traditional way is making them in solution. So just like
Hmm.
in a round bottom flask that you might imagine organic reactions to happen in.
Right.
That can be very useful, but it's hard to have that modular approach since you have to purify at every step. And so a huge breakthrough in peptide synthesis was doing the synthesis on a solid support. And so now it's solid resin that you can move around. The peptide is growing off of that, and you
Hmm.
can couple reagents in solution to that. And so, and then wash with organic solvents. And so traditionally, the types of synthesizers that you can buy commercially now are often batch based. And what that means is you have a reservoir of your resin that has
Right.
your peptide on it, and you add reagents in solution directly to that peptide, and you heat it and shake it for like an hour or 30
Hmm.
minutes, and then
Right.
you wash it away. So the new technology that was built in the Pentaludi lab is called continuous flow based chemistry. And so now instead of adding a bunch of solution to your peptide and then later washing it out, now we have a continuous flow of fresh heated reagent that goes through the resin bed and then continues
Hmm.
out as waste. And that really enhances the individual peptide coupling steps from maybe 99% to 99.95.
Right.
And that sounds like a very small improvement, but if you're doing 200 iterative couplings, that makes a massive difference.
Yeah, I can imagine. And I'm sure the work that you're doing right now has a very profound impact on drug discovery. And how do you see your work contributing to the broader field of organic chemistry?
Yes, so now I'm doing postdoctoral work with Alana Sheparts at UC Berkeley. And I'm still thinking about the chemical biology of peptides, but what my current work is developing a chemical reaction that forms a bond between two carbon atoms within the peptide backbone. And so if you think about peptides and proteins, that the bond that forms is between carbon and nitrogen. So that's called an
Hmm.
amide bond. Um, and in peptide and protein therapeutics, there's tons of amide bonds, but in some cases they can actually be liabilities. For example, your body can digest the amide bonds. Like you eat protein, you eat a hamburger, you
Yeah.
digest the protein. But if you're building a drug, you don't really want to digest it because you want your body to interact with it. And so I've developed a reaction that can basically replace that carbon nitrogen bond with a carbon carbon bond. And that's something that's really unique in nature. And it can be further derivatized into something called a heterocycle, which is really common in pharmaceuticals, but it's not very common within peptide backbones. And so it's a small step forward. We're developing new organic reactions, but the hope is that these can be applied, um, along the pipeline of actually making peptide therapeutics be more drug-like when you discover them, rather than having to make, a many mutations to them to make them more
Right.
drug-like down the line.
Right. So if I understand correctly, I think the sanctity of the peptides remains unchanged, but the amide bonds are now protected with a new combination of carbon-carbon, did you say, or carbon-nitrogen? Right.
Yes,
Okay. So
carbon-carbon bonds.
carbon-carbon bonds. And does that affect the effectiveness of the medication, or does it have any impact on the overall medication or
It
therapy?
certainly would. It certainly would. And so, sort of how I'm envisioning this, right now I've demonstrated this reaction on to happen once within a peptide.
Mhm.
So there's one bond that I can replace with a carbon-carbon bond and form one of these heterocycles.
Right.
Um, and so what I'm imagining is when you discover peptide drugs, you do an
Hmm.
initial screen. And so when you find a hit, something that could be your drug down the line, right now it's all amide bonds. And you have to do a
Right.
lot of what's called medicinal chemistry, chemistry
Hmm.
on that molecule to make it look more like a drug. But my hope is with my reaction, that can be included in the early phase. And so instead of looking at a molecule that's only amide bonds, you could look at a molecule that already looks just a little bit like a drug. And I
Mm.
hope that would it certainly enhances the chemical diversity that you now have access to, but I hope it shortens the time between discovery of a potential drug and finalization of the drug. That's my goal.
Right. Okay. Understood. Carly, you've mentioned the importance of mentorship in your career. Can you talk about some of the mentors who have influenced you and how they've impacted your work?
Yeah, absolutely. I've had a couple of research experiences when I was an undergrad that I think were very formative. Early on, I didn't really know what I wanted to do. I knew that I was really interested in health and disease. And so my first research experience actually was in disease biology. So I worked in a lab that studied Duchenne muscular dystrophy. And so
Hmm.
that's quite early on I was exposed to, that disease. Um,
Right.
and what we were looking at is phenotypes of early tissue to see if we could find earlier signs to diagnose the disease. But I was a young undergrad. I didn't really know
Mm.
anything about working in the lab. And
Yeah.
my mentor was always extremely patient and clearly generated a supportive, safe environment that allowed for a
Hmm.
lot of trial and error on my part. And yeah, that's really something that I try to emulate a lot. It's very clear, and now there's data to back this up, that if you foster a supportive, safe environment for people to work hard and take risks, um, that leads to growth of really amazing, successful, and also healthy scientists. So that was great.
Absolutely. Very well said. I think that's spot on, and it resonates with my experience too. And how you approach mentoring students and young researchers in your field, because I believe you work with students now, and you also mentor students in your field.
Mhm. Yes, I've had the pleasure of working with a couple undergrads at Berkeley, and that has been so much fun. I think all three of the students that I worked with, it was their first research experience. So a lot like my position in the story that I just described.
Yeah, right.
And my approach is really sort of like what they do in med school, where see
Hmm.
one, do one, teach one. So I will show my students a reaction, show them how to do something, and then I will ask them to do it, even if they're not very comfortable, but I'll be there to guide them when they ask questions or when they're doing something different. And then once they get a little bit more comfortable, it's clear that the best way to learn something is to teach someone else to do it. And so any
Right.
chance I get, if we have another new student or if someone has a question, I'll try and ask my undergrads actually to teach that person or to help that person. Because that sort of does two things. That helps solidify your learning in your own mind when you teach someone
Hmm.
else. And it also gives them confidence, like
Yes.
they're in a position where they're asking for a lot of help. And if you then help someone in return, you feel more like a part of the community and more like you belong in the lab, which is the case.
Yeah, absolutely. And I think the way you go through the material when you know you have to teach someone else is totally different from going through the material while you know that it's just for your own consumption. So I think the level of focus and the level of detail that you go into and retain changes completely and drastically. So that's a great method. And can you share an example of a successful collaboration that has significantly advanced your research?
Yeah, I mean, I think the best example is still our collaboration with Therapeutics.
Mhm.
Um, that really gave me a chance to think about academic research from a true applications perspective. I'm still in academia. I work at a university, and we're really interested in basic science. How does nature work? How does this reaction work? It's sort of rare that you can feel like you're having direct effects on society. And so I think that type of industrial collaboration can be really amazing for students, for graduate students. Um, yeah, I was really grateful for the ability to think about peptide sequences and what they can actually do and why people would care. And that really
Yeah,
changed my view of science and research in general.
right. Now, coming to challenges and innovations, what are some of the biggest challenges you've faced in your research and how have you overcome them?
Yeah, this is a really great question because I think the answers really depend on whether you're in an academic setting or an industrial setting.
Right.
So the challenge that really surprised me in my research, and especially during graduate school, is how long you could go without any good results. You know,
Yes.
research, especially experimental in lab research, is a lot of manual labor. And so you can put in countless hours for months and feel like you have nothing at the end. But then all of a sudden, you take an NMR and you look at the spots and you figure out like what's actually going on. And it's this whole moment of realization that actually, what you are doing is very cool, or like you found some new thing, or just finally you get out of that pit. But I think the types of valleys that you can enter where really nothing works for such a long time is
Yeah.
can be really draining. And that I was surprised, you know. I thought graduate school would be it's intellectually challenging, which it is. But the hardest part in the end
Yes.
was how much endurance I felt like I needed to have.
Wow. And what drives you to keep going even when things are tough or things get tough?
Yeah, I think still just the interest in, what can we find at the end? Like, and just remembering why I started in the first place. I really want to build molecules and to find molecules that can impact disease. And the deeper you go and the more you learn, the more you realize that we really don't know very much. You know, now
Yeah.
I'm really thinking about like what molecules already exist in the body that have an effect on disease, and we really don't know that much. And so that combined with knowing, like if you put in the work, if you do experiments, you will get data. Like data will appear if you put in the
Yeah.
work. And so you can always get out of that valley. It's just you never really know like how long it's going to be, and that is the challenging part. But if you just keep your head up, talk to your colleagues, ask lots of questions, learn a lot,
Right.
and you can also pivot if something is really not going well. You can put it on the back burner. I had to do this in graduate school. Put it on the back burner, come back to it later when you have a fresh mind, or when someone's said something really inspiring. You can always come back to it later.
Right. And honestly, I can't think of anyone who has never had a self-doubt. We all go through self-doubt. And what role does self-doubt play in your life and how do you deal with it?
Yeah, I'm sure you've heard of the term imposter syndrome, but
Yeah.
yeah, it can be really challenging, and I think having a supportive community helps a lot, but it can also be really challenging when you meet with or you interact with people who really put you down for one reason or another.
Yes.
Um, and I think that's where a lot of my self-doubt has come from. Um, but I'm grateful for lab mates and friends who have always been so supportive. And yeah, that's just the way
Yeah.
to get through it.
Yeah. I think in my case, I go through self-doubt quite a lot, and I just use it to uh, try to do better, and and that has helped me uh, shape myself into a better person and a better professional in many ways. So I think self-doubt sometimes can be good too. Uh, Kali, can you share an example of an innovative project or experiment that you've worked on?
Yes, let's see. So I think I'm really excited about my current work. We recently submitted it to a journal and it's on the archive right now. Um, and again, this is the reaction that essentially switches a native peptide bond with a carbon-carbon bond. Um, and I say like innovative experiment, but really it was, we had a hypothesis about what we expected should happen, and then that expected thing did not happen. And
Right.
so the innovation sort of just appeared, but you have to follow what the molecules are telling you. Um, and so instead of seeing, attachment of a second molecule onto the peptide backbone, I actually saw the peptide backbone itself just rearrange. So essentially
Mm-hmm.
there's two atoms where one was on the outside of the peptide switch to be on the inside of the peptide. And it was extremely unexpected. And so the innovation, I guess, was pursuing that really unexpected finding, thinking
Wow.
about this as something very important rather than just being a side product or some contamination that you should ignore. Um,
Right.
but I mean, it was an extremely serendipitous discovery.
Wow. And talking about the future directions and impact, what emerging trends or technologies in organic chemistry are you most excited about?
I think thinking about peptides as organic molecules is something that is extremely exciting. When I started my PhD, actually, people were really hesitant to say that peptides could be good drugs. You know, just as if you eat protein, you digest it, it gets excreted from your body. People thought peptides would just do the same. What's the point of taking a drug if you're just going to digest it? And so I think the field of peptides as therapeutics was really emerging 5 to 7 years ago. Um, and now Ozempic is a household name. So people are
Yes.
taking Ozempic for type 2 diabetes, but now it's also being widely used for weight loss. And that's a peptide. It's pretty straightforward. It's a linear, canonical, natural peptide with two unnatural modifications. So there's one amino acid that is unnatural to prevent digestion at that position.
Mm-hmm.
And then there's also a fatty acid chain. So like a fat-like chain on it that helps it
Yes.
kind of stay in your body for longer. And what I think the really crazy thing is that it has an effect in your brain, even though its
Yes.
target is near your gut. So
Yes.
people are realizing that this peptide is actually crossing what's called the blood-brain barrier, and it's having
Mm-hmm.
an effect neurologically. And I think that's extremely exciting because we don't know too much about how to design peptides that can do that. And yet we found one that has way more activity than we were expecting. So I think that's very exciting.
I think Ozempic is a breakthrough, and which is also surrounded by a lot of concerns and controversies, such as increased signs of aging and wrinkles and reduced muscle and kidney functioning or electrolyte
Yeah, because
disturbance and
there's
so
really a
on.
lot that we don't understand. And I think
Yeah.
it's sort of, and maybe it's not that rare, but it's certainly rare with a peptide that so many people would start taking it for its off-brand activity.
Yes.
Um,
Yes.
yeah, so it's exciting, but of course there's concerns. But what I'm excited about is people figuring out, what's actually happening. What is it really doing?
Kari, how do you envision the future of organic synthesis and its application in pharmaceuticals or other industries?
I think when people think about the future right now, it's hard to think about it without thinking about machine learning. And
Hmm.
so this is not my field of expertise, but people are really thinking about how machine learning can guide synthetic design. So you're never going to get rid of organic chemists. Like the intuition of well-trained organic chemists will always beat out machine learning models. But if these models can do two things, I think it would be really amazing. First is, can they help design what's called a retrosynthetic analysis? So if you have a very complicated molecule, what chemists do is they try to break bonds on paper to figure out how to get there. And there's countless ways that you could get there. Perhaps machine learning could guide that synthetic design. Like, what is the best route that's going to get the best yield? That would be great.
Right.
Something else that I know pharmaceutical industries are using right now is they're trying to use machine learning to determine whether a specific drug is going to have toxic side effects.
Mm-hmm.
So before actually doing clinical trials or even animal trials. But if you can screen out candidates that have toxicity, you can save a lot of time, money, effort, and also pain and suffering. So I know that's something that a lot of people are very excited about right now.
Right. So you would eliminate those options and not go ahead with any clinical trials and
Exactly.
just eliminate them early in the clinical trial phase, or maybe before the clinical trials.
Yeah. Mm-hmm.
Right. And what potential do you see for your research in addressing current and future scientific challenges?
Mm-hmm. Mm-hmm. I think what I've been thinking about lately is the power of unimolecular reactions. And so what
Mm-hmm.
that means is you have a reaction occurring within one molecule. And those are typically much faster than reactions that would occur between two molecules. Because if you have two molecules in space, they have to come
Mm-hmm.
together and do the
Yeah.
reaction. But if they're already together within one molecule, that reaction is going to happen a lot faster. And that's the reason why the reaction we discovered works so well. If that peptide bond was between two molecules, it would not happen. It just would be too slow. And so this is not a new field by any means, but I think if we're thinking about modifying peptide-like structures in proteins, thinking about these
Mm-hmm.
intramolecular reactions, I think will really open up doors to other types of weird modifications we could put on these previously biological molecules.
Right. Kali, what has been the most rewarding aspect of your career so far?
Mhm. I think the most rewarding part of my career is working with so many amazing people. I think
Yeah.
especially in the last couple of years, as I'm a postdoc, I'm really loving mentoring junior students and seeing
Mm-hmm.
them go through the parts of my career that I like loved so much. Just having like a eureka moment when you get your first piece of positive data and you feel like you're the first person in the world to learn this thing.
Yes.
I think that's just really exciting, and seeing, students gain the confidence that they can be scientists too. You can be an independent researcher. It's not reserved for, the high and mighty. If you're curious, if you have questions, you can get the tools to answer them. And
Right.
so one of my favorite parts has been mentoring students. But I also really love talking about science with others. I think the nature
Mm-hmm.
of chemical biology in general is extremely interdisciplinary. So, like I've mentioned, I've had the opportunity to work with experts in machine learning and biologists at pharmaceutical companies, and also
Mm-hmm.
really hardcore synthetic organic chemists. And they all have deep expertise that I certainly can't have expertise in all of those areas. But I feel like
Yes.
the ability to work well with experts in many areas and then have your own little niche of expertise is one of the coolest parts about, academic research.
Right. And and I'm sure this research work is very, very demanding. How do you maintain a balance between your personal life and research?
It's definitely a balance that needs to be maintained. Um, I think something that's the most important is having a network of friends and family who are outside of your bubble. So something we call like the academic bubble or the research bubble, um, to give you more perspective on the world and your life beyond the research that you're doing. Because often to you, if all you do and all you think about is your research project, if your project
Yes.
isn't going well, it's easy to think that your life isn't going well, and that's never the
Yes.
case. There's so much more to life than, you know, papers, awards, progress. And we have to remember kind of like why we're doing this and what we enjoy from it. And I've always found that maintaining connections with other people really helps me with that. And so specifically, I love playing volleyball. And so
Mm-hmm.
I've played volleyball throughout undergrad all the way up till now. And
Right.
I've met so many people who, I would never meet otherwise if I just, was at the university all day. So I think that's been super rewarding.
Wow, that's fantastic. And it's such a vicious cycle because you end up being friends with the people you hang out with the most, and you're
Mhm.
always at work, constantly interacting with people, and that becomes your bubble. And it's very hard to break out of that that circle and make friends
Mhm.
outside of work, uh, which is something I haven't been able to do yet. But
Yeah, it could
I
be
think
really
it's a good
hard. And,
practice.
yeah, it's also a blessing if you really love the people that you work with.
Yes,
And so I think
yes,
I've been really lucky. So many of my best friends have been, in the department or in my lab. And
yeah,
love talking with them. Um, but it's always important to maintain a perspective on the wider world because there's so many people who have no idea what you're doing and why. And so the other
yeah.
benefit of, having a network outside is you learn to communicate and think about why you're doing what you're doing and why it's actually important. Because if you can't communicate that well,
Yeah,
then that's a huge detriment to yourself.
exactly. Now, this is a philosophical question, and how do you define success, and has that definition changed over time as you grew and evolved into who you are right now?
Mhm. Yeah, I think that's a really great question, and it's something that everyone needs to define for themselves. And
Yes.
so it's something I've also been thinking about in terms of mentorship is, your goal as a mentor should be to help your mentee achieve their definition of success. But it's also important to figure out what your own definition of success is. And
Yeah.
I think mine has always been like, I really value learning new things, working with people, trying to make a difference. And that make a difference has really been what I think has changed over the years. When I started in grad school and I learned that I would be working with this pharmaceutical company, I
Hmm.
thought we could have a drug on the market. I wanted to work directly with patients, but
Right.
that's a much longer road than people expect. And so that's not something that can reasonably happen, when you're doing your academic training. Um, and I think I still have that goal. Um,
Hmm.
but I'd really like to come at it from an academic perspective. And I'd like to stay in the path between academia and industry. So I want
Hmm.
to do exploratory research, but I want the new things that we find to have implications for drugs and disease and health.
Right.
Um, and so that becomes a huge goal. And so I
Right.
think I've had to modify my definition of success to be, am I enjoying the research that I'm doing? Do I think
Hmm.
I'm having an impact? And is that impact positive on those around me? So the people that I work with, the people that I mentor, and then
Yeah.
hopefully down the line, patients. That would be the ultimate goal.
Right. And if you were to name a few people who contributed immensely to your incredible success so far, who would they be?
I think all of my advisors, um, I think especially during undergrad, as I mentioned, I went to an undergrad only college and I
Right.
worked in a research lab for my last year. And for part of that year, I was the only member of the lab,
Right.
but it's just crazy that I could have those resources and that experience. And so, Williams College definitely had a huge impact on me.
Right.
MIT, Brad Pinsaluti, like all the folks that I
Hmm.
worked there had a huge impact. And then, of course, I'm a fellow with the Miller Institute for Basic Research in Science right now. And so that's a special institute at Berkeley that funds fellowships for independent basic research in science. And so I'm
Right.
hosted by Alana Shepards in the chemistry department. But I
Hmm.
do get a little bit extra independence as a fellow to think about research and travel and communicate. And so I think really everyone that I've worked with throughout the years has been so immensely supportive and critical to my success. And certainly wouldn't be anywhere without all of them.
Right. Well, that's so well said. Are there any upcoming projects or developments in your research that you're particularly excited about?
Um, yeah, I think especially as I'm thinking about next steps,
Hmm.
I so I guess two things. My project right now, we've demonstrated this reaction happens in peptides.
Hmm.
Um, I'd be very interested to see if it could happen in cyclic peptides or proteins, or really other ways that we can access these interesting heterocycles that are embedded in the backbone. And so
Hmm.
I'm really excited about other reactions that could be done there. Um,
Right.
the other thing I'm excited about as I'm thinking about the next steps in my career are our bodies make really weird molecules, like really heavily altered peptides that are often really challenging to discover and are even more challenging to figure out what they do, you know, in our bodies and health wise.
Hmm.
So that's just something in general that I've been thinking about a lot and that I'm excited about.
Wow, what an insightful conversation we've had today. Carly, I want to take a moment to express my sincere gratitude for joining us on the show today. Your journey from academia to the cutting edge work you're doing now is truly inspiring, and I know our listeners have learned so much from your experience and insights today. Your deep dive into the chemical biology of peptides, the breakthroughs in your research, and the personal stories about mentorship and collaboration have painted a vivid picture of what it's like to be at the forefront of organic chemistry. It's clear that your work is not just advancing the field but also shaping the future of scientific research in profound ways. Kali, before we go, where can our listeners learn more about your work and stay updated on your latest research and initiatives?
Oh, yeah, sure. First of all, I'd just like to say thank you so much for having me on your show. This has been really fun, and I'm really looking forward to the other episodes that you're going to have. Listeners would be welcome to follow me on LinkedIn and also keep up with my Google Scholar. I think those are the best places to keep up with, what I'm doing next. So thank you so much.
It's a pleasure, Kali. It's wonderful having you here. And to all our listeners out there, thank you for tuning into another episode of Drug Diaries. If you enjoyed this conversation, make sure you subscribe, leave a review, and share this episode with anyone who's passionate about science and innovation. Stay curious, stay inspired. Until next time, this is Kaushik Trivedi signing off.